So in this video I will discuss on the class of carbohydrate, important class of carbohydrate, that is polysaccharides. Now the most complex and the most important class of carbohydrates are the polysaccharides, and these are generally called as glycans. Okay, now these glycans or polysaccharides, they are formed by the condensation of many monosaccharide units linked together through glycosidic bonds, as we have seen in the case of disaccharides and oligosaccharides. And these polysaccharides, they may be branched or unbranched, and they are divided into two broad groups: the homopolysaccharides, also referred to as homoglycans. Here in this case, upon hydrolysis, they yield only one type of monomeric units. Okay, and these are the examples: the cellulose, glycogen, and starch. And in case of heteropolysaccharides, these polysaccharides, upon hydrolysis, they yield two or more different types of monomeric units. Okay, example is hyaluronic acid, chondroitin sulfate, heparin, and murines. So these are some of the example of heteropolysaccharides. Now these polysaccharides, invariably they have very high molecular weight, and also they serve as structural components of the cell as well as functions as nutrients, and they can be hydrolyzed by acids or by enzymes, okay, to yield monosaccharide units or their derivatives. So now let us study these two important broad group of polysaccharides. So the first one is homopolysaccharide or homoglycans. Now, as I had mentioned, that these, upon hydrolysis, they yield only one type of monomeric units, monosaccharide units. Now one of the best example is starch. Okay, starch is the most important storage polysaccharide in plants, and starch, it consists of two types of glucose units. Okay, two types of glucose polymer, and those two types are amylose and amylopectin. Now a natural starch have about 10 to 20% of amylose and about 80 to 90% of amylopectin. So amylose, it is an unbranched chain of alpha D glucose. Okay, that means the chain is unbranched. It is a linear chain of glucose linked by the alpha 1,4 linkages, alpha 1,4 glycosidic bond, and this molecule, it contains about 200 to 2000 glucose units, which make a helical structure due to the bond angles. Okay, so it is just remember, it is unbranched and it is a helical structure which formed due to the bond angles, and the glycosidic bond involved is the alpha 1,4. That means it linked the carbon number 1 of 1 glucose with carbon number 4. So here many of these glucose units, around 200 to 20,000 glucose units, are linked together. So this is the structure of alpha amylose. Whereas in amylopectin, you can see that it is also a polymer. Amylopectin also is a polymer of alpha D glucose, okay, which are linked by alpha 1,4 linkages, but it differ in amylose in being highly branched. You can see this is a branch chain and this is the main branch. Now these short chains of about 25 to 30 glucose residues are linked in the main chain through alpha 1,6 linkage or through alpha 1,6 glycosidic bonds. Okay, because it linked with carbon number 1 with carbon number 6. Okay, so it is alpha 1,6 linkage. Now these bonds, these branches, they are linked to the main chain after every 20 to 30 residues. Okay, so amylopectins, they are therefore very large molecules with about 2 into 10 to the power 6 glucose units. Okay, so they are very large molecules. Now here we can see another example of the homopolysaccharides or homoglycans, that is a glycogen. Now glycogen is another very important storage polysaccharide in animal cell. Okay, and structurally, if you take a look at the structure of the glycogen, it is very much similar to that of amylopectin. That is glycogen. You can see it is a polymer of alpha 1,4 link subunits of glucose. Okay, and also with alpha 1,6 here, this link is alpha 1,6 linkage, and it forms a branches. Okay, so here the glycogen is a polymer of alpha 1,4 link subunits of glucose with alpha 1,6 link branches. But the difference is that the glycogen, it is more extensively branched. If you remember, here I said around after every 20 to 30 residues, but in case of glycogen, the branching occur after 8 to 12 residues. So glycogen is more extensively branched as compared to amylopectin, and it is more compact compared to that of starch. Okay, it is a glycogen is the structure of glycogen is more compact. So another example of homopolysaccharide is cellulose, okay, which is the one of the most abundant polysaccharide which forms as a structural component of the plant cell wall. And cellulose, it is an unbranched homopolysaccharide, okay, of about 10,000 to 15,000 of beta D glucose subunits, and these beta D glucose units, glucose residues, they are linked by the beta 1,4 linkages. Okay, beta 1,4 glycosidic bond. Thus, if you see the structure of cellulose, it resembles amylose, amylopectin, and glycogen, because those homopolysaccharides, they are also made up of the glucose units. But the only point of difference is that in cellulose we have the glucose is in, it has a beta conformation, whereas in amylose, amylopectin, and glycogen, the glucose residue, they are of alpha conformation. Now the polymers of beta D glucose, they are also represented as a series of rigid pyranose rings, as you can see here the figure below in the chair configuration. Okay, and they are connected by the oxygen atom bridging the 2 carbon in the glycosidic bond. Okay, you can see here in this figure, in which there is a free rotation, and each chair is turned 180 degree related to the preceding subunits, thus yielding a straight extended stable chair. This thus renders the fibers a great tensile strength, and part of a chain in which the D-glucose units are in beta 1,4 linkage. Cellulose, it cannot digested by mammals because of the lack of enzymes that cleave the beta glycosidic bond. Since alpha amylase, it breaks only the alpha bond, and even though it cannot be digested, but it plays a very important role in human nutrition. So this is another example of homopolysaccharides. Okay, so coming to the next important homopolysaccharide or homoglycan is inulin. Now, inulin is a homoglycan involving only fructose residues, and these homopolysaccharides, they are small polysaccharides usually with less than 100 residues, about 60 fructose residues. And this inulin, it is the storage product in some plants which do not store food as starch. Okay, so some plants, they store uh, the food in the form of this inulin, and this inulin polysaccharide, it is present in onion, garlic, asparagus, and roots of a number of genera belonging to Asteraceae family. And the anomeric carbon number 2 of the beta fructose is linked to the carbon number 1 of the next fructose, okay, by a glycosidic bond. So this makes an unbranched chain of fructose monomers. They are linked in this way. One fructose unit, the carbon number 1 is linked to the carbon number 2 is linked to the carbon number 1 of the next fructose residues. And these chain, they typically have a terminal glucose unit. As you can see, this is a glucose unit where the anomeric carbon number 1 of the glucose, it gets linked to the anomeric carbon number 2 of fructose. And this inulin sugars, they are usually sweet in taste. So another two important polysaccharides, they are dextrin and as well as chitin. Now dextrin is a nutritional reserved in yeast and bacteria, and it is also a homopolymer involving only the alpha glucose units, okay, as in the case of amylo pectin. It, however, differs from starch and glycogen in that in having the alpha 1,6 linkage as the main branch. Remember, in case of glucose, in case of glycogen and starch, we has one alpha 1,4 linkages as the main branch. But in case of dextrin, just remember the main branch is alpha 1,6, where the glucose units are linked together, where the anomeric carbon of one glucose is linked with the carbon number 6 of another glucose residue. And the branches here, they do not have any specific linkage, and any of the we can see any of the alpha 1,2, alpha 1,4, or alpha 1,3 linkages, they may be utilized, okay. As you can see here, in this case, the alpha 1,3 linkage also occur. Now you can see here the carbon number 3 of one glucose residue is linked with carbon number 1 of another glucose residue, okay. So in this case, it might link with carbon number 4 or carbon number 2 as well. So here the branches, they do not have any specific linkage, and any of these three linkages, they may be utilized. And dextrin is a bacterial product which is produced on the teeth surface, and it is also used as a food additive. Now another important polysaccharide, homopolysaccharide, is chitin. And here chitin is probably the most abundant polysaccharide of nature after cellulose. It is the principal component of the exoskeleton of insects and also of some fungi. Now, chitin is closely related to cellulose, and here the alcoholic group you can see of carbon atom number 2 in all these three, as you can see clearly, the alcoholic group of carbon number 2 of the beta D glucose unit is replaced by the N-acetyl amino group. Okay, this one, the N-acetyl amino group, and it is thus a linear polymer of the N-acetyl D-glucose amines units joined together by the beta 1,4 linkages. The linkage here is beta 1,4 because it connected the carbon number 1 and carbon number 4 of another glucose unit. And chitin's pronounced stability is based on the hydrogen bonding of the N-acetyl side chains. And like cellulose, chitin also is indigestible by the vertebrate animals. So these are some of the important homopolysaccharides or homoglycans. So in my next video I will show on the next group of polysaccharides, that is the heteropolysaccharides.